Hey guys, I miss you all so much. Oh, as promised, here is Troy Boy. So, this is my pupper, aka my child. I don't think he wants to get up now, but he's gonna say hi. Here's Troy Boy. Troy Boy, say hello. Say hello. He's grumpy because I woke him up from a nap, but this is Troy Boy. So, you get to meet my baby. So today we're just going to go over the DNA replication and protein synthesis prep test. So if you have your packet, this is going to be pages 16 through 19. And I want to do kind of how we do in class, where we go through the answers and why the answers are the answers. I think that that will be most beneficial for you guys. So I cannot write on my screen, so I'm just going to say the answers as we go. But it is important you follow along and you understand why the answers are the answers. Okay, number one, in mRNA, blank is used as a nitrogenous base instead of blank found in DNA. So in mRNA, you have the base uracil instead of thymine. So the answer would be A, uracil and thymine, because uracil's in mRNA and then thymine is in DNA. So one is A. Two, the codon AUG always signals what event in a cell? AUG is the starting codon. So that is the start codon. It's methionine. Therefore, that signals the start of protein synthesis. So 2 is B. Okay. The 3 stop codons, if you remember, you have UGA, go dogs, UAA, and UAG. So those are the stop codons, and then AUG is the start codon. 3, the enzyme blank unwinds and unzips DNA, so replication can occur. What unwinds and unzips? That is D helicase. If you put A, polymerase is what adds the bases, so DNA polymerase is what makes DNA. And if you're talking about making mRNA, RNA polymerase makes RNA. So what unwinds and unzips the DNA? That's D helicase. 4, what types of bonds hold amino acids together to make a protein? Okay, B, peptide bonds. Remember, because a protein is also called a polypeptide, and that's because the amino acids are held together with peptide bonds. Something to note is that it does say hydrogen bonds. What's held together by hydrogen bonds? The DNA or the bases in DNA. So if this is adenine and this is thymine, they're held together by hydrogen bonds. Okay, so hydrogen bonds is what holds the bases of DNA together. Peptide bonds is what holds amino acids together, and amino acids make up a protein, not DNA. 5, where does DNA replication take place within the cell? It has to be the nucleus, because DNA can never leave the nucleus. So if you're using DNA to make more DNA, it's always going to be in the nucleus. So 5 is B. 6, which of the following nucleotide pair bonds would be found in a DNA molecule? So what you need to know first off is uracil is not found in DNA, so that eliminates choice D. Then it wants you to figure out what pairs with what. Well, adenine pairs with thymine, cytosine pairs with guanine, and the only one of those that fits is B, so 6 is B. 7, the backbone of a DNA molecule is made of which 2 components? This one's kind of hard, but you should definitely know it. So think of what a nucleotide's made of. A nucleotide's made of 3 things. It's made of a 5-carbon sugar, a phosphate group, and a base. The base is the rungs of DNA. So the other 2 are what we call the backbone. So that would be C, phosphate group, and deoxyribose sugars. Now, what if it said phosphate group, or what about choice A? Choice A says phosphate molecules and ribose sugar. Well, I mean, it's made of the 5-carbon sugar and the phosphate, so why is that not the answer? Well, it's because DNA has deoxyribose as its sugar, whereas RNA has ribose as its sugar. So 7 would be C, not A, because it doesn't have ribose, it has deoxyribose. That's why it's D in A. 8, ribosomes are made of A, rRNA and protein. So remember, rRNA stands for ribosomal RNA, tRNA stands for transfer RNA, and mRNA stands for messenger RNA. So the only one of those that makes up ribosomes is rRNA and some proteins. 9, Watson and Crick were the first to suggest that DNA is B, the shape of a double helix. And again, remember that they used Rosalind Franklin's work. Don't forget her, because everyone wants to forget her, and she's important. Okay, so 9 is B. 10, the process by which a DNA molecule is copied is called, okay, C, replication. Okay, when you are copying an exact copy of DNA, that's DNA replication or DNA synthesis. If you put D, translation, no, that's when we're using the mRNA to make a protein. That's a completely different process. So with DNA replication or DNA synthesis, we're literally just copying DNA, and we have to do that to go through mitosis. If you remember during the S phase, DNA is copied. That's what DNA synthesis is. So that and then translation are two completely separate things. 11, a DNA nucleotide may be made up of a phosphate group along with, okay, so the two other parts for a nucleotide. So nucleotides, you have a phosphate group, a 5-carbon sugar, and a base. If it's DNA, our 5-carbon sugar has to be deoxyribose. And since it's DNA, the 4 bases we can have are adenine, guanine, cytosine, and thymine. So that eliminates A. Yes, it would be deoxyribose sugar, but there's no uracil in DNA. So A is out. B, ribose sugar. Nope, it has to be deoxyribose sugar. Okay, so B is eliminated. C, deoxyribose sugar and thymine. Both of those fit. And then D says ribose sugar. It's got to be deoxyribose. So 11 would be C. 12, which series is arranged in order from largest to smallest in size? Okay, this one threw me up because I did not read the question. Can you believe, can you imagine I had the audacity to do that? Yeah, and I'm pretty sure about half of you did too. So you're doing largest to smallest, not smallest to largest. So the smallest one of these, or the largest one, I did it again, gosh darn. The largest one of these would be, let's read what we have. Chromosome, nucleus, cell, DNA, nucleotide. All of those things are part of a cell. So cell would definitely be the biggest thing. So since cell is the biggest, that eliminates A and C, because we know cell comes first. Okay? B says nucleus is the next largest. D says nucleotide is the next largest. Well, what is a nucleotide? It's the building block for DNA or RNA, building block for nucleic acid. So that's definitely not the largest. Okay, the nucleotide is your phosphate group, 5-carbon sugar, and base. That's definitely not second largest. So the answer would be B. Cell is the largest, then you have the nucleus, then you have chromosomes, which are in the nucleus, then you have DNA, which is wound up to make chromosomes, and then you have a nucleotide, which is the monomer of DNA. 13, X-rays, ultraviolet light, and radioactive substances that can change the chemical nature of DNA are classified as, these are all going to be D, mutagens, because they mutate your DNA, and they can cause mutations, and so that's, and through those mutations, they can cause cancer, and that's why they're bad. Okay? I really like these three. It's having you think kind of outside the box, and I love it. So 14, applicants for the 4th job of the help wanted ad in table 11.1. So it's this supervisor one right here. Must be able to follow exact directions from double-stranded template. That double-stranded template eliminates RNA because RNA is single-stranded. So that's telling you directions from DNA because DNA is double-stranded, RNA is single-stranded. Travel from nucleus to the cytoplasm. And you know it's not DNA because DNA can't leave the nucleus. So what is 14 talking about? Here are our 4 options. We have DNA. Nope, DNA cannot leave the nucleus, so that's eliminated. mRNA, that's a possibility. C, tRNA. tRNA doesn't do anything with DNA. And then rRNA doesn't do anything with DNA. So 14 is B, mRNA. Remember, mRNA is made from the DNA and then leaves the nucleus, goes to the cytoplasm so it can be translated. 15, third job, must be able to maintain genetic continuity through replication and control center activity by regulating or regulation of enzyme production, limited number of openings, all benefits. And then I also want you to pay attention to the title on that one. It does say executive position. So in the cell, what is the control of the cell? DNA. Okay, DNA is like the control center. It maintains everything that's going on. So 15 is A. And then 16, applicants for the second job, vital in the field of translation. So we know it's not D, or it's not DNA because it's not vital in translation. DNA is not involved in translation. Applicants must demonstrate skills in transporting and positioning amino acids. So what transports amino acids? tRNA. So 16 is C, tRNA. Okay. 17, which structure shown in figure 11.3 is a pyrimidine? You don't need to know that, so you can go ahead and skip that one. 18, which structure shown in 11.3 does not contain a nitrogenous base? You need to know this. So 18 is basically asking which of these does not have a base. If you look at A, that is the base guanine, so A is not a choice, not the correct choice. If you look at B, that is the base thymine, so that's not a choice. If you look at C, that's the whole nucleotide, and part of the nucleotide is the base, so that's not correct. D is the phosphate group of a nucleotide, and it does not contain a base, it's just the phosphate group. So the answer to 18 is D. 19, what type of mutation has occurred here in figure 11.4? If you just looked closely, it kind of gave you a huge hint. You have the initial one up here, and then when the mutation happened. So if you look right here, what's that word? It is deletion. Okay, if you're deleting a base, that is a frame shift mutation. So 19 would be B. And again, frame shift mutations, insertions and deletions are really, really bad because if you look, it has changed every single amino acid after where that mutation occurred. And that's really, really bad for someone because they can't develop that protein properly, and that could be really, really harmful. What will be the result of the mutation in figure 11.4? So a frame shift mutation. A will have no effect on protein function. It will definitely have an effect. The protein will not be able to function. Only one amino acid will change. No, if you look at it, we went from phenylalanine to leucine. We went from glycine to alanine. We went from alanine to histidine. We went from leucine to cysteine. Okay, it changed every single amino acid. So 20 would be C. Nearly every amino acid in the protein will be changed. You don't put D because you're not certain that the organism would die. If it's not a protein that's super critical for functioning in life, they probably wouldn't die. However, if it was one that was critical for functioning or for life, they probably would. But it doesn't guarantee that they're going to die. Figure 12.5 shows the structure of, so it's wanting you to look right here. It's wanting you to look at the whole thing. Sorry, the whole thing. So what is that? I see two strands. I see the bases C, G, A, T. So both of those things tells me it's DNA, not RNA. It's not single-stranded, and I don't see uracil. So what is X labeling? Let's just ask that one. What's X labeling? So X is labeling the nucleotide. Here is the 5-carbon sugar. And again, with DNA, that 5-carbon sugar is deoxyribose. This is the phosphate group. Both of these make up the backbone of DNA, which is just the sides of DNA. And then the third part of the nucleotide is the base. So here we have cytosine. What do these lines represent? These represent the hydrogen bonds that hold bases together. Very important you know that. And you also need to know what pairs with what. So if I gave you a test and I had this base blank and I had C right here, you better know that G goes with it, guys. That's easy peasy, free points. So take advantage of that. 22, DNA is copied during a process called, is copied during a process called replication. A. Transcription and translation you're not copying DNA, you're making mRNA to then make a protein. Okay, so 22 is A, DNA replication. That's when we're copying the DNA. 23, and eukaryotes' DNA is, please get those correct. Oh my goodness.